Mendel had all the valid reasons to. His theory is correct, and it's actually Mendelian genetics is. We still study Mendelian genetics, and this is how all our genetic disorders we are able to understand. So it was not Mendel's problem. Yes. Good evening, Atesh. How are you?
Fine.
So we are discussing chromosomal theory of inheritance today. And the question is, what do you already know about Mendel? Why was Mendel's work not recognized for a very long time? What were the reasons?
Yes, sir. So there was a lack of communication during that time.
Yes. So
then, like the idea of genes, factors, the idea of genes and factors were not clear at that time. Then the mathematical approach for biology was not accepted. Yeah, these are the reasons I remember.
yes, so the major reason is that the way he did his study, he included mathematics to understand and explain biological phenomena, which was a new thing. And scientists and biologists of the time were very skeptical of his mathematical reasoning to explain the biological phenomena. Second thing, yes, the communication was not that good. So it was not the problem of Mendel's work. It was the problem that it could not get communicated to a broader scientific community all across the globe, so that people could critically think over it and also gather evidences in its support or against it. So that was the other thing. And as you said, that genes were not very well understood. Okay, so these concepts were not very well understood at that time. And Mendel, being the pioneer of genetics, that's why it's known as the father of genetics, he brought upon the biological world a very new thing, which people were not very, very aware of. Okay, so these were the reasons that his work stayed unnoticed. It was not out of spite or people didn't like Mendel or something like that. They just could not understand the work at that time. And the work also could not reach the broader audience to gather support or critique. Then when it was recognized, so who were the people and how was it recognized later on? You remember people? Anyone would like to comment on that?
Sir, Von Schermark and I think Cody or something like that. I don't remember the name. I'm not sure.
Hugo de Vries. Remember Hugo de Vries?
Yes, Hugo de Vries.
So they independently figured out Mendel's work very late. So people, whenever scientists get stuck somewhere or they want to know some better perspective of any work, so they always dig into literature. They try to see, and that's one of the backbones of science, that things should be documented. Okay, so science is documented. Whoever has done anything in science should be replicable. So if it is true logical science, there should be a protocol to do this, and anyone who follows the protocol should be able to replicate the results. That's what science is. Science is not like magic, that it works with someone and does not work with other persons. Given everything and every parameter stays the same. So they were looking, they were reading for their own work, and they came across Mendel's work, and they realized that Mendel have tried to answer some very fundamental questions of inheritance, how characters are transferred from the parents to the offsprings. And later on, that work was popularized again, and Mendel's work again came back to the center stage, and people now, because it could now reach a wider audience, by that time people now started reading and critiquing or accepting the work. So when it got accepted, there were two scientists, Sutton and Boveri. So now coming to chromosomal theory of inheritance, they realized that what Mendel said, so Mendelian principles of inheritance actually holds true and can also be understood in the light of chromosomal segregation. So the way Mendel talked about these genes and or these factors, we now know due to our cell biological advancement in microscopy and other techniques, that this is how chromosomes also get segregated. So this chromosomal theory of inheritance is a theory that united the knowledge we had about Mendelian principles of inheritance and the knowledge that Sutton and Boveri gathered about chromosome segregation and its transmission from one generation to another through the gametes. Okay, is that clear? I think, was it Hanifa?
Yes,
Who was it? Yes, Hanifa. Is it clear to you?
yes.
So the exactly what they mean was, let me go back to this diagram. Yes, this diagram. So Mendel talked in terms of capital T, small t, capital R, small r, capital Y, small y, right? And Sutton and Boveri figured out that one chromosome can contain either a capital T or a small t or another like capital T. So only one. So this one arm, so this is one chromosome. These are two sister chromatids because they are replicates of each other. So, but one chromosome will have capital T. The other one coming from the other parent will have a small t or a capital T, depending on what is coming from the other parent. But one chromosome only carries one allele. So basically, this inheritance happens through chromosomes, which people earlier did not know. That what is the actual thing, what is the physical signature of this inheritance. So the physical signature is this chromosome. On this chromosome are these alleles, which are the different forms of the same gene, slightly different forms of the same gene. Okay, is that clear, Hanifa? Any other doubt associated with this?
No, sir. Thank you.
Yeah. Yeah. So, and also then Thomas Hunt Morgan, as we discussed, he experimentally verified this theory. Okay. So the chromosomal theory of inheritance, he experimentally in Drosophila, he did genetic experiments. And the good thing about Drosophila is you should know is that Drosophila only have 4 chromosomes. Okay. 4, like n equal to 4. So a haploid Drosophila has 4 chromosomes. So the gamete of Drosophila will have 4, out of which chromosome, so there is one X chromosome. Okay. X or like Y. So there is one sex chromosome and other 3 are autosomes, out of which one is very, very small. So it's not very significant in terms of genetics. So you're left with 3 major chromosomes. So it's easy to do genetic experiments on Drosophila and prove that as chromosomes segregate, characters also segregate, and inheritance happens in a Mendelian manner. Okay, so chromosomes, segregation of chromosomes follow Mendelian principles. So this chromosomal theory of inheritance was given by Sutton and Boveri, experimentally verified by Thomas and Morgan, and it is based on the Mendelian genetics of inheritance. Okay. Make sense, everyone?
Yes.
Any other doubts, people, please? What else do you want me to go over?
Linkage and recombination.
Sorry, Hanifa?
Linkage and recombination.
Linkage and recombination. Okay. So in continuation, let's discuss this. So what is linkage? Okay, Mendel gave 3 laws. Okay, so I'm going through a quick revision, and this is what these classes are for. So you people have to answer now. So tell me the 3 laws of Mendel. One person will tell one law and explain that law. Yes, Hanifa, let's start with you.
The law of dominance was one of his laws.
Yes, what does it say?
The factors occurs in pairs, and there's always one dominant character.
There is one dominant allele for that character. Okay.
Yes.
So yeah, so character can be height, and height can either be short or tall in pea plants where Mendel did the experiment. So Mendel said that for every character, there are two possible scenarios. Okay. For a flower color, it could be white or purple. For a height, it could be short or tall. For the position of the flower, it could be axial or terminal, etc., etc. And he said that when these two alleles for the same gene are present together in a heterozygous condition, one always dominates over the other. Okay. Which means that, let's say capital T is for tall height, and small t allele gives rise to small height. So when capital T and capital T, both parents will give capital T, for sure the height is going to be tall, correct? When both parents will give small t, for sure the height will be short, because these are homozygous conditions. But in a heterozygous condition where there is a capital T and a small t, what will happen? According to Mendel, what will happen in a heterozygous condition? Yes, Fatima. Fatima or Donna? Anyone?
Neither that tall nor that short, like in medium.
It will be medium?
Like the height, capital T and small t, right?
Capital T and small t will be?
It won't be that tall nor that short.
So logically you will think it won't be that tall and it won't be that short. But if that is true, then there is no dominance. So law of dominance, then Mendel will not give law of dominance. Instead, what Mendel found is that it's exactly opposite. Whenever capital T and small t is present together, the plant is always tall. There is no medium-sized plant in P. So the character has only two extremes. Either it will be tall or it will be short. There will be no medium. So that's where he gave this theory of dominance, and which works with most of our genes, except for some exceptions in nature. Exceptions are rare. Exceptions are less than 1% of what's happening in nature, where biology is trying to get out of the box and getting finicky and doing all sorts of things. Okay. So there are very few rules in biology that cannot be altered at all. Okay. And as we are discovering more and more, and that's true in general for universe as well, for physics, chemistry, anything. Biology is known as science of exceptions, but I think every subject, every field of science is like that only. So if you talk about gravity in physics, you talk about the speed of light in physics, or you talk about certain reactions being spontaneous or non-spontaneous in chemistry, they may have exceptions at different regions of the universe or in different conditions of a reaction. So similarly, biology is a living laboratory of nature. So there are certain exceptions. But overall, the broader thing is whenever any of the two genes, so for example, we always have either, for example, we have black, one type of hair color, right? Let's say there's a parent, a couple, and one parent has black hair, the other parent has blonde hair. So you will never see like that the kids are having one black, one blonde, one black, one blonde, or a mixture of half black, half blonde hairs. Either it will be black or blonde, correct? It could also be something entirely different from the parents, but most of the genes, what I'm trying to say, works on the law of dominance. So one allele is dominant over the other. So when they both are present together, only one expresses. That's the law of dominance. Okay, Shaheen. So capital T small t will be a tall plant. Yeah.
Yes.
And we call that kind of a tall plant. What do we call that kind of a tall plant? We call it a hybrid tall or a heterozygous tall. Okay. I think these things, you should revise these things now. So this was the first law. Are there any exceptions of law of dominance? Donna? See these classes which we have, yes.
Yes, sir.
Are there any exceptions of law of dominance?
Not sure.
Think about it. I told you about law of dominance. And do you remember studying anything about, so law of dominance says that one allele is dominant over the other. Are there systems where this is not followed?
Yes. Isn't
Yes.
codominance an example?
Yes, codominance is a exception of law of dominance because there is
Yes.
dominance of two alleles, equal dominance of two alleles. And where do you find this happening in biology?
Think AB blood group.
Exactly, in our own bodies, in human blood group, right? So the AB blood group is only possible because of here, codominance. So in codominance, both alleles in a heterozygous condition express themselves equally. So there could be, so anyone would like to explain this quickly? How about you, Donna? Because you gave this example. So would you like to explain what is on the screen quickly?
I just know that AB blood group is an example, but I'm not sure how to explain it.
Okay, so like, do you know what is ABO blood group and how the blood group works?
No.
No? Okay, so this could be one of your doubts, right?
Yes, sir.
Anyone who would like to explain this to Donna? And while explaining, you will get your concept strengthened. I'm sure that someone of you might be knowing how to explain this because this could be one potential 3 or 4 marker question. And we have done it in detail.
I can try.
Yes, Anifa, very good. Perfect, go on. All you have to do is to try, that's all. We all are doing the same.
So the ABO blood group in humans, it is controlled by the gene I.
That's not the gene. It is controlled by the genes for A antigen and B antigen. I is just a representation. IA means the gene carrying the gene for antigen A. IB means the gene for antigen B, and IO means this gene, like there is no gene for antigen A or B. IO itself is not a gene. So IO means nothing. So either you have that antigen, okay, from your parents, you get that antigen, or you don't get that antigen. Okay, so
Yeah.
yeah. So there are only two antigens, A and B. O means nil, no antigen. Yes, now explain.
So there are 3 alleles for this IA, IB, and I.
IA, IB, and IO.
IA, IB, and IO.
Yeah. Also in books, sometimes written as IA, IB, and small I.
IE and IB they're dominant over small I, but like IE and IB together, they're co-dominant.
Yes. How can a person have a blood group? How many blood groups are possible in humans? Hanifa?
Yes. A, B, B and O.
Yes, A, B, and O. Correct. We are not taking into consideration the RH sector here yet. So forget about that, Abhi, for the time being. So you are correct. There will be 4 blood groups that are possible. And how one can have these 4 groups? What genetics need to be there? So if someone is having blood group A, what could be the genotype? Possible genotypes?
It could be IA and IA.
Mhm. That's it. Can it be IAIO?
It can be IA and IB.
Yeah, because you yourself said very correctly that even when it is IAIO, A can be dominant over O, so only A will express, right?
Yes, sir.
Similarly, what are the conditions of genotype in which a person can have a B blood group?
IB and IBIO.
IB and IBIO. Yes. But then I think that it's a perfect example of dominance, no? Why do we call that the ABO blood group is codominant, shows codominancy? You know, I'm just trying to ask trick questions. So tell me. People, come on. Why is ABO blood group called an example of codominance, where we can see that A is clearly dominant over O, B is also dominant over O, and O can only express when it is present in a homozygous condition. So the blood group ABO only shows codominancy in AB blood group. So when antigen A, the gene for antigen A, or the allele for antigen A and the allele for antigen B is present together, then there is a dilemma. None of these, either A or B, can express themselves over the other one, and there is a codominancy. So both express equally, and as a result, the blood group can either be A or B. So it becomes AB. Is it clear?
Yes.
Yep. So that's codominance, which is one of the exceptions of law of dominance. Is there any other exception of law of dominance, Atif?
So in incomplete dominance,
Yes, and what is that? What happens in incomplete dominance?
like both the genes are not equally dominant. When both the genes come together in a heterozygous condition, it's like present a mix of both their phenotypes.
Yes, so this is exactly what Hanifa was talking about, right? Was it Hanifa or was it Shaheen? I'm a little confused, but either Hanifa or Shaheen. You said that when capital T and small t will be present, there will be a medium height, correct? So that's not true in the case of pea plants, but it has been seen in case of the flower color of the snapdragon plant, where there are two alleles again, capital R and small r, denoted as capital R, small r. So these two alleles, one allele produces the red pigment, which gives the red color to the flower. The other one does not produce that red pigment and the flower color is white. Now, following the laws of Mendel, we will assume that if there's a capital R, homozygous condition for any of the allele, a homozygous condition for capital R or a homozygous condition for small r, of course, the flower will be pure line red and pure line white respectively. But in a heterozygous condition, you will expect the flower to still show a red color because R should be dominant over small r. But here it does not happen. Here there is a blending, and due to that blending, the flower color becomes pink. Okay. So none of these alleles, no single allele is able to show dominancy, and both the alleles end up being incompletely dominant over the other. Okay. Is that part clear, everyone?
Yes.
Yeah, cool. So first law of Mendel is done and two exceptions of it is done, right? Now, second law, anyone? Hiba, would you like to speak? You are not participating. I hope your microphone is working. Is it? Do you have any doubts? Please bring all your doubts in today's class, and then we will do questions related to genetics unit, previous year's questions. But to do that, you need to solve all your doubts first. So which is the second law of Mendel? You can stay silent like this. I have no issues, but this class is for you. Or I can, yes, Rifat.
Law of segregation.
Law of segregation. And what does that law says, Rifat?
Alleles, they don't show any blending. And there is the gametes, they receive one allele, which gets segregated independently.
So you mean to say that it says segregated in what? Where is it getting segregated? Where is it going after segregation?
To the gamete.
To the
Gamete.
gamete. Perfect. So you know the answers. I don't know why people are just hesitating and not speaking it up. Okay. So cool. So it goes into gamete, which means you're saying that for every character where the alleles are in pair, wherever the gametes have to be formed, each allele segregate and goes into one gamete. So one gamete only receives one allele for every character. Is that what you're trying to say?
Yes.
Great. And that's exactly true. So this is what law of segregation is. And do you know what it is also known as?
No.
What this law is also called as, anyone? The other name for law of segregation of gamete? The law of segregation? I'm sure we have written it during the class, so please go back and revise. And also you can write it again. It is known as law of purity of gametes. Okay. So second law, if I go back and talk about the law. So law of dominance is here. And it's down. Yeah. Law of segregation, it's called law of purity of gametes. And this is a universally accepted law and it has no exceptions. Okay. Why do you think law of segregation have no exceptions? Or do you think that we have not found an exception yet? And are there chances that we can find an exception later on? High chances. Of course, there are always chances of things happening in biology because it's life. Life is only predictable to a certain limit and degrees. But why is this law without exception? Why is it universally accepted law? This was a legit question that can come. The question is why, which law is a universally accepted law and why? People, anyone? Fatima? Yes, I think Fatima is. Yes, Hiba, we're talking about law of segregation, but why it cannot have any exceptions is the question. So which law is universally accepted? Of course, it's law of segregation as it is written here. But why it is universally accepted? Why it has no exceptions? The reason is gametes have to be maintained as uncontaminated entities of inheritance, right? So if there is blending happening due to incomplete dominance, right? So here I have shown you all the three parameters. In a pea plant where there is a complete dominance, in a snapdragon where there is a blending, and in blood where there is a codominancy. Whatever be the status of these alleles in a heterozygous condition, whenever they will go and form gametes, this capital T will go in one gamete and small t will go in another gamete. And even in case of snapdragon, where capital R and small r together make pink flowers, when there will be gamete formation, capital R will be one type of gamete and small r will be another type of gamete. Like one type of gamete will contain capital R allele and other types of gametes will contain small r allele. And if this capital R, which came from a blended parent now, finds another capital R in the progeny, and the progeny has capital R, it will again give rise to red flowers, which means that even though there was a blending happening at the morphological level, at the level of gene function and gene expression, the allele itself remains pure and untouched, and it cannot show blending whatsoever. Okay, so when they will get segregated, they will be as pure as they were before they came and blended in this parent. Does it make sense, people?
Yes.
Yeah. Okay. What is the third law? Anyone? Third law of Mendel, and what does it states? Fatima? if we'll quickly get over with this understanding, then we'll start solving some questions in today's class. Are you there, Fatima? Am I audible, people?
Yes.
So anyone would like to explain the third law of Mendel? Because connected to that is recombination and linkage, and then we can finish this part and can go for some questions, important ones. So third law.
Is it law of independent assortment?
Yes, it is exactly the law of independent assortment. And what does it say, Donna?
It says that when two pairs of traits are combined, one pair, segregation of one pair of the characters independent and the other than of the other pair of characters.
Yes. So other pair of alleles, okay? Or so character is one. Character, so we are talking about two characters here, a pair of character. For example, height and color. Okay, these are two characters. Each character will have two alleles, correct? So this law says that one character, so it says the alleles of two or more different genes get sorted into gametes independently of each other. So it talks at the level of genes, not at the level of alleles. If the segregation is happening at the level of alleles within the same gene, that is law of segregation, and it is a pure law. If I'm talking about segregation of alleles of different genes, then it is called law of independent assortment at the genic level. Is it clear? So in short, it says that if a parent has black hair and black eyes as well, and another parent has a blonde hair and brown eyes, okay, let's say. So it's not true that the kids will either have black hair and black eyes or brown hair and brown eyes. So when gametes will be formed, the black hair character, the alleles for black hair, will segregate independent of the alleles of black eyes. So these two are two different characters, and their alleles will segregate independently of each other. Is it clear, everyone?
Yes, sir.
Yeah. So you should also work and pay some attention, all of you. And how are you going to write the answer? The better you explain in the most concise manner, the more marks you get at board. So this is how boards work. Yeah. Okay. So, but this law is not universally accepted. So we know that this law has some exceptions, and the exception here is linkage. Now, which was the original question asked by someone, that can we go over linkage and recombination. So let's first talk about what do we mean by linkage. What is a linkage? Yes, so I come here. Linkage. So linkage is an exception of law of independent assortment. Can anyone tell me? Can anyone explain linkage quickly? No one? Okay, so linkage was a term which was coined by Thomas Hunt Morgan. And when he was studying his Drosophila for the genes and the inheritance of genes, he did some similar kind of experiments that Mendel did in pea plant. And since he was doing it in animals, like in Drosophila, so he realized that every time when you take two genes, so one for yellow bodied, one for white eye, you take two characters together and study their inheritance, they do not follow the Mendel's ratio in the F2. Okay. So in F2, if you remember, for a dihybrid cross, the ratio should be 9:3:3:1. Correct? I'm sure all of you are clear with how to make these dihybrid crosses. Yes, no? Am I audible, people? Am I audible? Because no one is speaking, so.
Yes.
Yeah, okay. So he realized that, so my question was, I hope you all understand and know how to make these dihybrid crosses and get these ratios, right?
Yes.
Yeah.
Yes, sir.
Yeah. So in the F2 generation, in a dihybrid cross, following the Mendelian genetics, these four characteristics, 2 in one and 2 in the other parent, follows these 9:3:3:1 ratio. Okay. But when Morgan did it with particularly these two traits, the white body and yellow body and white eye of one parent with brown body and red eye of the other parent, he realized that this ratio is not followed in F2, which means somewhere down the line something is wrong. Why are these characters not segregating like Mendel expected and Mendel said that they will segregate? So the reason is there was a physical association which is known as linkage between these two genes, between the genes of body color and eye color. And you will find that if you take a yellow bodied fly, which is also white eyed, then more progenies will have yellow body with white eyes. And so either progenies will have yellow body, white eye, or brown body, red eye. Very, very few progenies will have a brown body with a white eye or a yellow body with a red eye, which is recombinants. So you find less recombination in some particular genes. Now, these are the genes where law of independent assortment does not hold entirely true all the time. So they are called linked genes. There are many linked genes that were discovered after that, after Morgan's discovery of this. And Morgan also gave this relationship between linkage and recombination, where linkage is inversely proportional to recombination. So higher the linkage, less the recombination. If there is 100% linkage, let's say theoretically speaking, if there is 100% linkage between two genes, what percent of recombination will be there? Yes, people. If there is 100% linkage between two given genes, what percentage of recombination will be there? Yes, Rifat, can you hear me now?
Yes.
The question is, if there is 100% linkage between two given genes, what percent of recombination will be there? There will be 0% recombination. Okay. And how do we find linkage? We find linkage as physical association. So if, let's say, on a chromosome, this is a chromosome. Okay. In this chromosome, there are, let's say, there is this gene A, this gene C, this gene B, then a gene D. Okay. And let's say this is the distance is 3 units. Okay. The distance between A and C is 1 unit, and between B and D is 4 units. Decreasing linkage. Which gene pair in the order of decreasing linkage, which means which will be the highly linked genes? The first. The question is for Donna. Yes, Donna.
Could you repeat that?
Yeah. The whole point or the question? The question? Yeah, the question is, you have to arrange these gene pairs in the order of decreasing linkage. So high linkage to low linkage.
Isn't the one
There are 4
one-
genes. Your voice is very, very feeble, Donna.
Usually closer has high linkage, if I'm not wrong.
Yes, correct. So which pair will have the highest linkage?
One.
One is, this is 1 unit of distance
Okay.
between the genes. Genes are A, C, B, and D.
So it'll be B.
Will be AC. The pair AC will have the highest linkage. Gene A and gene C.
Okay. Yeah. AC.
Yeah. Gene pair. Yeah.
Then AB.
Then.
AB.
Are you sure? That's where you will do some silly mistakes in entrances if you don't pay attention. Look here. The distance between A and B is 3 units.
Yes.
Distance between A and C is 1 unit. So what is the distance between C and B?
2 units.
2 units. That will not be given to you. You have to just figure it out. So AC will have the highest linkage, then comes
CB.
CB, correct. Then
AB.
AB.
B, yeah.
Then
and BD.
BD and
AD.
Mhm.
CD
CD,
and then AD.
then AD. Is it clear, everyone?
Yes.
Yeah.
Yes.
And if I ask you, arrange them in the order of recombination, so it will be exactly opposite. Correct? Because linkage is inversely proportional to recombination. So can you arrange the genes? And I'll ask the question. So what will be the arrangement in terms of recombination? Arrange these gene pairs in the order of recombination from talking about low recombination to high recombination. Atif. Yes, Rifat, tell me. Lowest recombination to highest recombination.
Will be AC, CB, AB, BD, CD, AD.
Yes, so it will be the same. Why? Because I have switched this order. I'm asking low recombination to high. So the one with high linkage will have lowest recombination. So the order will remain the same. Is linkage clear to everyone?
Yes.
Yep.
sir.
Okay, great. Now let's come to, so anyone who would like to just explain me first this, not the crossing over, just this diagram. What do we mean by homologous chromosomes and what I have shown here? Can you speak, Fatima? I don't think Fatima can speak. So anyone? Atif, would you like to explain what's homologous chromosomes? People? Rifat, Heba, Shaheen. Just a quick recap of what is here on the screen. No one would like to give a try? Okay, so Fatima is having some issues with the mics. It's okay. I think many of you are also struggling with network issues. Yeah, that I can understand, but try to be on a good network if it's possible, people. But these classes otherwise are of like will make no sense if you people are not if you're not discussing. Okay, it will be again then me giving a repeated lecture on the same topic without knowing that what actually you need. So your feedback is very important. Your discussion is very important. Your participation in the class. So question is, what are homologous chromosomes? Then we can finish a recombination also today and go to some questions. So homologous chromosomes are the same chromosome which is inherited by an offspring from the two parents. Okay. So for example, chromosome 1. So as I told you, there are different number of chromosome pairs in different organisms. Like in Drosophila, there are 4 chromosomes. In humans, there are 23. chromosomes in the haploid cell, respectively. So let's say chromosome 1 of a human. So every human gets chromosome 1 in pair. One it gets from the mother and the other it gets from the father. So for every chromosome, there is a pair. Now these two chromosomes, which are pair and basically are the same chromosomes, they have the same length, the same centromere position, the same staining pattern, and the same number and location of genes. Is this clear?
Yes.
Yeah. And on every chromosome, there'll be one. For example, here, there is a capital T allele. So on the other one, there will be another allele. It could be capital T or small t. I'm just making it different. So I've taken small t. And for one chromosome, during DNA replication, this one arm replicates to give rise to two arms. So if there are two arms of the same chromosome, they are called sister chromatids. And if I'm talking about two arms from two different chromosomes, okay, if they are in a homologous chromosome also, they are called non-sister chromatids of a homologous chromosomes. Okay, make sense?
Yes.
Yeah. Now in every chromosome, so if there is capital T here on this arm and small t here on this arm, what will be present at the same position, let's say here and here? In the chromosome 1, which we are getting from the mother and the father, on the sister chromatid, which allele will be present? It will be the same allele, people, because the chromosome got replicated. It's a copy of itself, its own arm. It's one chromosome only with two arms copied. So this will be capital T and this will be small t. Similarly, talking about this R, so here at the same location will be a capital R, but in a homologous chromosome at the same location, there will be a small r, because this chromosome has a small r allele. Is it clear or?
Yes.
Yeah. Okay. Now every chromosome has two arms on the opposite sides of the centromere. So centromere position tells what type of chromosome it is. Okay, there are types of chromosome on the basis of centromere position, which I'm giving you as a homework today. We have studied it, but go back and write down. You have to look at what are the types of chromosomes. What are the types of chromosomes on the basis of the centromere position? Okay. So if the centromere, which in normally it is not present exactly in the center, so it's present slightly offset of the center, which leads to the formation of one bigger arm and one smaller arm. Okay, so the bigger one is called Q arm and the smaller one is called P arm. And I told you that the P arm, the P stands for the word petite. Petite means small. Okay. So sister chromatids, non-sister chromatids, everything is clear. And the position of the gene on a chromosome is known as locus. So gene locus. So if I say gene locus, it means where is that gene located on that particular chromosome. Okay. So capital R, capital T, all these are the locus of that allele of the gene. Is that clear, everyone?
Yes.
Yeah. Now someone has to explain me recombination. So this is where I have talked about formation of recombinants, this figure. Who will explain recombination? Come on, I urge someone to. They just looks. Don't get don't get intimidated by this image. I'll be here. So someone should just lead, and if you are not able to, yes, Atif, please.
Yes, sir. So, like recombination is the formation of non-parental combinations due to the crossing over of chromosomes.
Perfect.
Yeah. So the crossing over of chromosomes happen placed during meiosis, which forms the chiasmata. Then the chromosomes which are crossed over are known as the recombinant the chromatids which are crossed over is known as the recombinant chromatids, and the ones which are not crossed over known as the non-recombinant chromatids.
So chiasmata is formed between what? Between which chromatids? Sister chromatids or non-sister chromatids?
Non-sister chromatids.
Non-sister chromatids, right? Is it possible that it can be formed between sister chromatids?
No.
It does. It's very rare in biology, but NCERT says no because not to add complexity, but it is not entirely impossible. It is possible, but if there is a chiasmata formation between sister chromatids, what will happen? Because this question has been asked in entrance. So there is a chiasmata formation between the sister chromatids of homologous chromosomes. So what will be the outcome? You will find more recombinants, less recombinants, no recombinants at all, or none of the above.
Can you repeat the question?
Okay, the question is, if chiasmata formation, write down, just write this question. If chiasmata formation takes place between sister chromatids, what will be the likely outcome? What will be the likely outcome? Option A, you will find more recombinants. Option B, you will find less recombinants. And option C, you will find no recombinants at all. And option D, none of the above options are correct. More recombinants, less recombinants, no recombinants, none of the above options are correct.
So it also depends on the linkage, right?
It also depends on the
Linkage, like the distance between the genes.
Yeah, so it depends on the linkage for like recombination between those two genes will be possible or not. but recombinants can be formed, you know. So let's say linkage will just tell that, let's say T and R, there are less chances of T and R getting segregated, more chance of R and Y getting segregated. There's no change. Let's say linkage, there is no linkage, let's say. No linkage between any genes. Will your answer change if I talk about linkage?
So is it more recombinants?
What do others think? How many of you support Atif's answer? Raise your hands. That there'll be more recombination. How many of you think otherwise? So give answers. This is where we are discussing questions. So look at everything is in front of you on the screen. Just look there and tell me the answer. You have written the question. Everyone should write their answer in the chat box then. If you are too shy to speak, then please write your answers in the chat box. So Atif, I know Atif says more recombination. Atif, you can also look, and if you wish, you can change your answer. And if you think the same answer, you want to stand with the same answer, write it in the chat. Shine, you have to type it, because the options that I gave, I'm not sure which was C. Just say more recombination, less recombination, no recombination at all, and none of the above options are true. So more, less, no, or none are true, something like that you can write. I just received one answer till now. Okay, Atif, am I audible to you?
Yes, sir.
So you're not giving your answer in the chat. Donna,
I would like to
Fatim.
stay with the same answer, sir.
Yeah, so you mean more recombination. Donna?
Yes.
Fatima, Hiba. Okay. Fatima? I see very less enthusiasm today. What happened? You people got bored of biology or what? Fatima, we are still waiting for your answer. Are you there? Okay, I'm not sure if Fatima can hear me. Yeah, so the right answer is there will be no recombination at all. And I got 2 correct answers, but one was not sure. So there's no, you have to just look here, you cannot be not sure in a exam, right? Whatever you will write, that will be counted as your sure answer. Because if it will be correct, you will get the mark completely. If it will be incorrect, your marks will be deducted completely. So why there will be no recombination at all? And if it makes logical sense, then it is the correct answer. You have to be sure about it through logic. There will be no recombination because, look here, if I make a chiasmata between these two from here, let's say, so the same sister chromatids get entangled among themselves, and this part goes here and this part comes here, it still will be the same combination, small t, small r, small y, small t, small r, small y. Even if there is exchange between the same chromosome, it will still be small t, small r, small y, small t, small r, small y. Do you understand, people? Atif, do you understand? Yeah. So chiasmata or crossing over between sister chromatids does not make any difference in terms of the gene recombination. Okay. So that's why what matters for recombination is the crossing over bridge or the chiasmata formation between non-sister chromatids. Next question is, how many chiasmatas can be formed between 2 homologous chromosomes? Only one? More than one? More than 2? No, more than 2 doesn't make sense. So more than 1 is 2 or more than 2. So 1, more than 1, or can there be no also? Let me give you 2 options, 1 or more than 1. Tell me. How many chiasmatas can be formed between 2 homologous chromosomes? One, two, multiple.
Not sure.
Yeah. Not sure. Okay. What do you think? What's your guess? What will you do when you will not be sure in an exam? You have one option always to leave that question, but you cannot keep leaving the question, right? That's not helping. Of course, doing it wrong is more damaging, but leaving is also not helping. So you have to try to get to some sureties through concept. So have you people started revising at all? How many of you have not started revising genetics unit or reproduction unit or biology as per? Raise your hands, those who have not started revising due to any reason. So you all have started revising. Okay, that's cool. So let's do one thing. I still find that you people need to revise genetics with some more dedication. So let's do this activity. I'm giving you genetics to revise. In the next class, I'll solely dedicate to questions from all the chapters of genetics. So you have time tomorrow. Day after tomorrow, we'll meet again. And when we'll meet again, we'll just take questions from the genetics unit, chapter 5, 6, and 7. Cool? And everyone should get their microphones fixed. Otherwise, there's no point joining the class, because if you are just going to be silent, then I've already covered the syllabus, right? So I'm just taking these classes because it's important for you to revise with me. The syllabus is covered, people. So be more vocal, come with some doubts. If you have no doubts, then answer in the class, because this is your opportunity, the golden opportunity where you can make mistakes, learn, solidify your concepts. That will help you in entrance and boards both. Okay, being silent is never helpful. Okay, so let's end this class on this note, and please revise, and let's take the next class as an opportunity to do as many questions as we can, one after the other, from the whole genetics unit.